Um, Sir, this is the last like 15 minutes. Um, I was recording a video, but my phone died and everything was lost. So here is the introduction. This article is important because it helps scientists learn about plant genes and how flower colors can change. It studies morning glory flowers because their colors are easy to see and help attract insects that move pollen. The scientists who wrote it work on the University of California, Irvine. I chose this article because I want to learn how plants grow and how things in the environment can change flower colors in different ways. Here's a slide. Sorry again. Methodology. I learned that the researchers tested 9 Ipomia species based on flower color, which is also called anthocyanin pigment. I also learned that they studied 2 Ipoporia plants, one normal and one mutant, to help understand how pigment genes work. To do the experiment, oh wait, my charger just flew out. To do the experiment, sorry sir. To do the experiment, they placed the flowers in greenhouse conditions. They collected different flower parts like the limb, tube, anthers, and stigma from open flowers. Then they froze these parts in liquid nitrogen so they could extract the RNA for testing. Let me check my battery. Okay. I learned that they organize our DNA sequences using computer software like Fred, Frab, Conced, and Polyfred. They also use PALP to make phylogenetic trees to see how the genes are related. Then they extracted total RNA using a method, from O'Neill and ran it through a gel to separate it. They transferred it to filters, checked the RNA amounts, and washed the samples carefully. Finally, they used gene-specific probes for CHSD, CHSE, DFRB, and CHI to study the genes. Oh, and again, there is the slide. Methodology 2.4. Lastly, the flower tissues were fixed, embedded in paraffin, and then cut into 10 microgram sections. Digoxygenin labeled RNA probe system was used to cut the sections. CHSD and DFRB were labeled with both sense and antisense probes. Localization was done with a Leica DMR microscope with Nomarsky optics. The results: the paper reported that most of the differences in flower color among morning glories are due to how genes for pigments switch on or off, not mutations in the genes themselves. The scientists made this discovery using gene expression comparisons and a statistic of the DN-DS ratio. The gene expression test evaluated a range of the kind of activity being exhibited by the genes in different species, and DN-DS ratio was about whether or not the genes coding parts were withstanding natural selection. Okay. And results. Okay, again. I learned that this ratio shows how genes change over time because of either small DNA changes or silent changes. This helps show if a gene is evolving normally, being removed through natural selection, or changing in a helpful way. I learned that pigment genes had different activity levels between species, but the actual gene parts stayed mostly the same. The DN/DS test showed no strong signs of major positive selection, meaning the genes are very stable. Overall, I learned that the differences in flower color are mostly caused by changes in how genes are turned on or off, not changes in the genes themselves. In the discussion, I learned that chloroplast DNA in plants slowly changes over time. This is important because it helps scientists understand, I don't know how you want me to present, understand how plants evolve. The results show clear patterns because many plants were compared. I also learned that chloroplast genes stay mostly the same because they are important for photosynthesis. And I think more plants or different DNA regions could be studied to see how changes happen over time. And that is my presentation for molecular evolution of the chloroplast DNA sequences.